So the paper that we're focusing on is called Cyclin Synthesis Drive, Early Embryonic Cell Cycle, and we are focusing on figure 3D. The major question for this article is cyclin enough to drive the cell cycle into the mitotic state in an early embryonic cell? We see that cyclin is an embryonic protein that accumulates during interphase and has a rapid decline during mitosis. Throughout the study, cyclin is a mitosis regulator. The mRNA of cyclin is studied and is suggested to induce maturation in Xenopus oocytes. These oscillations of cyclin in relation to the maturation promoting factor and H1 kinase leads us to believe that cyclin might have some role to play in catalyzing nuclear breakdown, chromosome condensation, and the formation of the mitotic spindle. The importance of the cytoplasmic factors of H1 kinase and cyclin. After observing the proteins that are involved in cell cycle and what happens, we are able to see a trend between the MPF, H1 kinase, and cyclin. To be able to observe the effects of these proteins while they are measured, Xenopus oocytes are used. Can you go back? Cyclin is an embryonic protein that accumulates during interphase and helps the oocyte reach maturation. In this experiment, cyclin is used from a sea urchin to see whether if cyclin alone will be sufficient to induce the oocyte. H1 kinase is important for the stabilization in the structure of DNA and chromatin, transcription regulation, and chromosome spacing. MPF importance. By observing the cell cycle, the question about what drives the cell cycle comes up, and if there is a biological clock within the cell itself. Every cell has 4 stages in the cell cycle, as seen in the figure to the right. We have G1, which is cell growth, the S stage, which is the DNA replication, the G2 stage, which is the preparation for mitosis, and lastly, cell division. By answering the question, we have to take a look at some other factors that have an influence on the cell cycle. MPF is just before the mitotic stage because it leads to the cell into nuclear breakdown, allowing to go into cell maturation and separate into 2 sister cells. As we see in the figure below, the cytostatic factor will stabilize the MPF, causing it to go into arrest throughout the second phase of meiosis. CAF is a calcium sensitive, causing the stabilization of MPF. Now we will look at the model organism. The Xenopus lepus is an African clawed frog which is used as a model organism in this experiment. Amphibians are used because they have large transparent eggs that allow for observation within the development of the embryo. This model organism allows for large eggs to be observed and manipulated while they mature. Since their development is rapid, that makes it the perfect organism along with their large eggs.
So how are we going to approach the research question? Well, we know that all cells go through cell cycle regulation. And knowing that they go through cell cycle regulation, we can also manipulate that process of cell cycle regulation, which consists of protein synthesis and the overall induction into the multiple interfaces. So what we're going to do is we are going to analyze cell cycle regulation in vitro cultivations of oocytes eggs. And through this, we are going to manipulate that process and try to reveal cyclin's significance within cell cycle regulation. So this leads us into our workflow and how exactly we're going to accomplish this approach. Well, first off, we need to cultivate these extracts in vitro of these oocytes eggs. So in order to do that, we need to centrifuge the 3 samples of the eggs that are provided. And we need to wash it in an ionic buffer that will replicate the composition of the cytoplasm of the ascites eggs. Now, when you centrifuge something, you allow the innards of the egg to spill into that ionic composition and thus kick off that process of cell cycle and cell cycle regulation. So once we find that our extracts mimic the exact cell cycle regulation of in vivo eggs, we can then use that as our basis of manipulation for when we introduce that pancreatic mRNAs, eliminate the endogenous DNA, and reintroduce that cyclin mRNA in order to determine if it does have any significance in inducing mitosis. Um, so how are we going to track cyclin? Because you can't just do that and not know where cyclin is coming from or how much cyclin is present in the cell cycle. So these extracts will be coded in S35 methanine, and that is a radioactive version of the amino acid methionine, which is actually the first amino acid present in protein synthesis. And when you introduce that radioactive version, it's going to bind to the proteins that are being synthesized. And what's that going to do? It's going to turn the proteins into radioactive molecules. Or yeah. So once we uh, uh, eventually track the rate of the cyclin through the addition of S35 methanine, we're going to extract it and uh, arrest the extracts at each process of the cell cycle through that electrically activated in vitro extract culture. And then we take the proteins out and we lay them on a radioactive polysaccharide gel. And what that does is that when you combine that radioactivity, you're going to get these black bands that indicate nuclear morphology. And the more, radioactivity you see in these bands, the greater the presence of cyclin. And that's how we're going to track cyclin throughout the entire process of this. Is this S30 methanine? Um, so this leads us into our figure, which is the presence of cyclin within these extracts that are mRNA dependent because we eliminated their endogenous mRNA. And so what we can ask ourselves when we look at this figure is, was cyclin enough to complete the cell cycle? And it's what correlation does it have with the H1 kinase and the maturation proliferating factor? So let's see. So in order to do that, we took our extracts and like I said, we eliminated the endogenous DNA with the pancreatic mRNAs. Then we added that mRNA SYN that inhibited the pancreatic mRNAs from, further decomposing any other, from further halting protein synthesis. And then we added the methionine, and we were able to gauge the um, presence of cyclin. So
So here we see that we have our controls. So the extract that was used to observe and manipulate the oocyte, this is what we have. And then by doing this, the time that is recorded for each of the phases will become the control to be able to reference and check the levels of each protein. It will allow us to do so. So by having this as an observation of an oocyte that was not disturbed, it would be the control for this figure. So on the next slide, we see that in this figure, a sea urchin mRNA cyclin dependent extract was added. This mRNA extract was significant in great detail. So when we look at the figure, there's a very defined proportional relationship between the H1 kinase, the cyclin, and the MPF, which is the maturation promotion factor. So we see here in our graphical representation in our figure, we see that at 40 to 60 minutes of timing, as well as 100 minutes of timing, there is a very sharp decrease that occurs during the NBD, otherwise known as the nuclear breakdown. So this also signals that there is a decrease at the very tail end of mitosis. So moving on to our conclusion. So our conclusion derived from the article pretty much consists of confirming the role that cyclin plays in the activation of both H1 kinase as well as the MPF, the maturation promotion factor. Based on the data from the study, we can see that cyclin follows an oscillatory pattern that moves in conjunction with the nuclear breakdown. So definitely we have identified the correlation between those two topics. So this, along with the fact established that mitosis cannot occur without the introduction of cyclin, mRNA, into the dependent extracts. So this helps us further answer our research question. So in conclusion, and to restate the main goal of this presentation, cyclin is very much essential to the induction of the mitotic process. Additionally, the interplay between these three components displayed a correlation when one followed a downward trend, and so did the others. So finally, on the next slide, we will talk a little bit about our critique on both the figure as well as the experiment presented as a whole. So yes, as a group, we do extensively support the conclusion that has been proposed above. So obviously, cyclin has been a very important vehicle for the cell cycle in this research article, and its multiple associations to both the MPF, the maturation promotion factor, as well as the H1 kinase and the proven details leave us with a very clear view, actually, of the importance of cyclin in the schematic process of the cellular cycle, leaving us knowing that cyclin is absolutely necessary for the cell cycle. Thank you. We have enjoyed this presentation, and here are our references. As you can see, we have the article here, and thank you very much for watching our presentation. On behalf of Dianna, Rayyan, and myself, Francisco, we thank you for your time, and we hope you enjoyed this presentation and it was of academic value to you. Thank you.